Good morning. Uh, I'm very happy to be here with you in this virtual makeup class. Thank you so much for joining. Uh, to miss a class, your class is not a single class, actually, couple of classes, 180 minutes, in fact. So it should be covered by a makeup class. This is why I tried to arrange this makeup class and you all cooperated. And thank you so much. Uh, Tomorrow we shall have face-to-face class in person. And tomorrow you can see your uh, evaluated answer scripts, midterm, as well as the quiz exams. Uh, Today we shall discuss some new topic and obviously tomorrow we shall continue it. In your curricula, so many contents are suggested on abiotic stress tolerance as well as some other uh, contents not so up to date. I am not sure whether this course uh, was previously offered by someone or not. But anyway, considering the course title as well as your degree, I shall try to give you the up-to-date science, obviously in line with the content of your course. Uh, Tomorrow, I would like to start at 6 p.m. if it is convenient for you. Winter is becoming a bit uh, severe. So the earlier we can start would be good, but it depends on your convenience. Uh, what do you think? Can we start at 6 p.m. tomorrow? Do anyone have any inconvenience? 30 minutes earlier start of the uh, routine. If not, then it's fine. Tomorrow we shall have a short session of showing you the answer scripts and then we shall go on. Today, let me show you today's topic. It is an interesting topic because uh, the breeding for is it visible to you?
Yes, sir.
Okay. So what you can see here, can anyone tell me what is visible?
So speed breeding for climate smart, abiotic, and biotic stress tolerant crop variety.
Excellent. So today we shall talk on speed breeding. Breeding is an art of genetically modifying the uh, plant or any other organism to improve the uh, yield or stress tolerance or any other traits. So speed breeding, uh, why speed breeding? Speed breeding actually to speed up the process of breeding because classical breeding to develop a new crop variety takes a longer time and it is a big problem obviously. So speed breeding can save the time. Let me tell you first the the content. Sustainable development goals to zero hunger. United Nations declared 17 sustainable development goals to be achieved by uh, 2030. One of the number one is that is poverty-free world, that is no poverty, and the second one is zero hunger. Uh, and to achieve this goal, we need to produce more food, uh, significantly higher amount of food that we are producing now, and to improve the yield. The land is, limited resource worldwide. So what we need to do, we need to develop a smart variety that can tolerate the harsh environment and can give us higher yield and better nutrition. So speed breeding is necessary to overcome this process because, as I mentioned, classical breeding takes a longer time. And there are some historical breakthroughs in plant breeding and the concepts of speed and precision breeding. Another term is precision breeding. So precision breeding and classical breeding, there are some differences. For example, gene editing is a precision breeding, very precisely. Even recombinant DNA technology we use for development of genetically modified uh, crop plant is also a precision breeding. Very precisely, you change the trait of a plant by altering the genetic sequence, either insertion of a new gene or silencing, deletion, or making any correction in the wrong DNA sequence. And we shall also discuss the strategies of speed breeding. How can we do it? And uh, today I shall coin the new term to you, gene editing. For example, CRISPR-Cas. I am not sure you heard about this term, CRISPR. Clustered regularly interspace short palindromic repeats. Uh, and Cas CRISPR associated uh, protein or enzyme. And this is a system discovered in prokaryotic organisms like bacteria and archaea. And this system is an adaptive immune system in those organisms. And that system evolved in the prokaryotes to defend themselves from the attack of virus. So it is a sort of natural molecular scissor, and scientists um, since 2012 they started to use this technology for editing the genome of living organism, and it's really a revolutionary technology which has been recognized by the Nobel Committee, and two female scientists, Jennifer and Emmanuel, received Nobel Prize in chemistry for this revolutionary technology. So we shall introduce it, and tomorrow we shall discuss in detail about the genome editing or gene editing. Now, question is why food security is a challenging task? Why SDG 2 is zero hunger, that is to make the world free from hunger. What is the current situation of food security in this world? The reality is that currently we have 8.2 billion people on this earth, and it has been estimated that nearly 2 billion people are going to bed with hungry stomach, and hungry people are angry people. You cannot think of any peaceful world with this situation. So, food security, obviously a challenging task. It is not a new challenging task since, the history of agriculture started about 10,000 or 12,000 years ago. Food security has been a challenging task in all ages. It is not only the less production on this earth, but equal distribution and natural calamities. Lots of factors. Some are natural and some are man-made uh, that are impacting on the uh, unequal distribution as well as the insecurity of the food. Climate change poses increasing risk to crop production through droughts, salinity, uh, high temperature, low temperature, extremely low temperature, different kinds of insects, diseases. In agriculture, especially the crop production, you may have a very good variety, but as we grow the crop plants uh, you know, outside the house, that is not control environment, as we are living, they are exposed to the uh, natural environment. So natural calamities sometimes create serious problem. For example, flood, or if there is no rain or very high temperature. And you know that global climate change is now a big concern and one of the most talked issue. And agriculture is obviously vulnerable to the global climate change. The different stressors of climate change, like I mentioned, drought, salinity, pest attack, they are increasing, and it is a big challenge. How can we adapt to the changing climate is really a big question, and your subject is associated with that focus. We need to develop or genetically modify the crop plant so that they can tolerate uh, or resistant to the different stresses due to the global climate change. From 1981 to 2000, scientists studied in a science paper uh, on rice, maize, and wheat varieties that had been improved through traditional breeding, boosted crop yield by 22 to 46% in Asia and Latin America. That means breeding, traditional breeding significantly contributed to increase the yield of productivity of the crop plants in Asia and Latin America, even worldwide. But the bigger question is due to the uh, population increase. Currently, we have 8.2 billion people on this earth. It is estimated that by 2050, the world population would be 9.7 to 10 billion. And to feed those, increased population, a global increase in crop production uh, should be 100 to 110% from now or 2005. It was a study. Currently, we need at least 70% higher yield than we are currently have. In Bangladesh, by 2050, it is estimated that the population would be around 250 uh, or 230 million. So in that case, obviously we need to increase our food production. The biggest limitation of food production is that horizontally we cannot increase the land area because it is a strategy to increase the land area to increase the total production. But another strategy is to go vertical because horizontal expansion, especially in Bangladesh, is almost impossible because every year we are losing the agricultural land, cultivable land by nearly 1% or like that. Even the real statistics is maybe more. So land is shrinking, population is increasing, and global climate change is posing a new threat. This year we experienced a severe flood in several districts that destroyed the crop production and also affected the human life directly. So, in this scenario, we must be smart enough to uh, do the breeding effort uh, faster as we did in the past. We have no choice. So improved crops are urgently needed. Improved means uh, those are tolerant to the uh, adverse environment. For example, we can say climate smart crop varieties are urgently needed to meet growing demand for food and address the changing climate. So it is now a global consensus, and it is very, very important for Bangladesh. In the Western countries, especially the developed countries, population are declining. So they do not care about, the increasing of the food. But in our case, uh, we badly need effort for increasing the productivity as well as smart technology for higher yield. So if we look at the global scenario, as I mentioned, 2050, the world population would be 9 billion, 9.7 billion, nearly 10 billion, but uh, uh, 2100, 10 billion plus. So it is impossible to feed the growing population unless we have uh, new technology. Can we produce sufficient food from zero point 0.2 hectare? That means zero point 0.2 hectare per person worldwide average land area. So food security is a formidable challenge worldwide, and the citizens of world must work together for a hunger-free, more peaceful world using the best conventional crop technology and the best of biotechnology in policy framework conducive to crop production. That means we need the smart technology like biotechnology. Let us look at some other sustainable development goals that are also linked to the or related to the crop production. For example, end of poverty for all. Most of the farmers, they are poor. Uh, they are resource poor, they are poor in different aspects. So improvement of agriculture, crop productivity is also one of the ways to end the poverty of a large number of people on this earth, even in Bangladesh. In Bangladesh, even currently, nearly 50% of the workforce are directly, indirectly related to the crop production or agricultural activity. Second, I mentioned freedom from hunger. Yes, and hunger, achieve food securing and improve nutrition and promote sustainable agriculture. This is the greatest target, and we are uh, focusing in this course how by designing a new plant, changing the genetic makeup, we can contribute to the uh, food security. Another uh, goal number 12, sustainable consumption and production. Ensure sustainable consumption and production patterns. Actually, sustainable consumption, food waste, we need to, reduce. Food waste is a big issue. If you go to the um, any party, you can see lots of foods are waste. So we must be very careful. And sustainable ecosystem agriculture plays a vital role in you know uh, local and global climate. So we use lots of technologies in agriculture. For example, chemical fertilizers, pesticides, they are creating severe pressure to the environment, and environmental pollution or other hazards, they are threat to the biodiversity. So, and degradation of the land as well as degradation of the soil health. Soil is the basic, natural resource for the existence of all biomes. So we must consider the soil health. So in that case, smarter agriculture or advanced agriculture must be, focused the issue of sustainable ecosystem. That means our ecosystem, we should maintain its productivity uh, for the future. Otherwise, it would be hazardous for future generation. I discussed possibly this slide timeline of key plant breeding techniques and technologies. It is believed that agriculture, the age of agriculture, is about 10,000 years or more. And domestication started uh, 10,000 or 12,000 years ago. So over the years, um, uh, human effort to make higher productivity, ensure food security, and human addition of human curiosity to improve the to understand the uh, mechanism of plant growth, yield, as well as the uh, biology. Gathered lots of new knowledges and lots of breakthroughs are in we experienced in the agriculture over the centuries. First, 1760, first hybridization performed. Hybridization means crossing of two dissimilar plant. Actually, naturally, by bees, different insects, and other organisms, pollination are taken place because plant cannot move away; they are sitting dark. So they need uh, some agents for their for transfer of the male, pollen from to the female, and this is the natural genetic modification of the plant, but it is uncontrolled. When the crossing of two dissimilar plant artificially you do, then you can get a hybrid, the F1, the next the seed developed and they are F1. often scientists found that F1 gives higher yield potentials than the both parents, and it is called heterosis. So hybrid varieties are now cultivated vegetables mostly worldwide, even in Bangladesh, hugely being cultivated. In early 80s, people were against hybrid varieties, but now no problem. But hybrid varieties giving us some heterosis added value, increasing the uh, yield as well as they are resistant to some of the stressors. That means combining the uh, uh, good traits of two dissimilar parent, we are getting the hybrid. In case of hybridization, a big limitation is that if a farmer uh, gets the seed, uh, he cannot get the hybrid seed of his own and cannot continue. So depend on the industry. But anyway, it is contributing the higher yield of our country. If you see 1856 selection theory been developed. Selection theory means to select the best, fruit or best plant uh, and get the seed for the next season. And actually, without any classroom, farmers, growers, they adopted this one and they found that the good seed from a good plant giving the good yield. But a big revolution taken place after two giant scientists. One is Charles Darwin. He has given a theory of evolution, how plants are evolving or organisms are evolving uh, through the ages. Evolution means genetic mutation naturally even taken place through the selection pressure uh, in the environment and also some other reasons. So at that time, no gene or no molecules are being discovered, but that insightful, idea as well as evidences, observation gave us a good clue for now we are doing the precision breeding. So 1866, Gregor Johann Mendel, he did a fantastic experiment. He was not an agriculturist or scientist. He was a priest, and he did an experiment with garden pea. And he developed two laws how during crossing between two dissimilar plant, the genetic traits are transferred to the next generation. Until now, Mendelian laws are very, very crucial for designing a breeding program. And then you see 1903 pure line theory. Pure line theory means if your line, that is plant line, is pure, genetically homozygous, then you can get homozygous plant. Otherwise, segregation for the next generation, if you grow dissimilar plants, you will find and it affects the yield. Bulk selection breeding, new technology developed, recurrent selection, pedigree and back cross breeding. These are the smart classical breeding technology being developed, and around 1928 mutation breeding, that is you can artificially make evolution, that is mutation in the genome of a plant by using mutagenic agent, maybe chemical or the gamma radiation or ion beam. And although mutation breeding is not precision, but if you do it on the seed or plants, you can see due to the injury in the DNA, you can find the variation in the trait in the later generation, and you can pick up some of the bad gene. If mutated, then you can get the a good trait or like that. Loss of function, we can say, loss of bad function or like that. So gradually recombinant DNA technology and already you studied. So they came and colchicine. Have you heard about colchicine chemicals which can uh, uh, cause the doubling of chromosomes? So in uh, classical breeding, colchicine treatment also changing the chromosome number or ploidy level by using chemicals. So this is very powerful mutagenic agent. uh, People also developed some variety. And single seed descent, that is selecting a single seed from the best plant and then gradually get the a series of generation to find the best plant. Reciprocal recurrent selection, type breeding, lots of breeding technologies, but one of the greatest uh, revolution, we can say, is called the Green Revolution. And the father of Green Revolution, perhaps you know his name, Professor Norman Borlaug. Uh, Professor Norman Borlaug, he got the Nobel Prize, Peace Prize, for his revolutionary work. He developed, dwarf variety of wheat because he found that some of the plants in the field they are very short, but he worked in Mexico, but they are uh, tolerant to the lodging and yield. They are also disease resistant. So dwarfing related gene has been a big target, and in rice and other crops it has been introduced, and yield was significantly increased. In Bangladesh, first such variety was um, IR8. It was from the ERI, came to Bangladesh, and yield increased several fold. Previously, our rice varieties was very long, and they are prone to the losing. But uh, recent varieties, most of them you can see they are short, straight, erect. Leaves are getting more sunlight, more photosynthesis, giving the yield. And then, all these I mentioned, they are, classical breeding technologies. But last century, first recombinant DNA technology when uh, you know uh, uh, was developed. That is, a foreign DNA we can transfer from one organism to another. Foreign DNA means from one species like bacteria. From a bacterium, if there is a good gene, you can pick up it, clone it, and then transfer it into any DNA of any organism, and really amazing and revolutionary. And we call the changed organism GMO, genetically modified organism. Flavor saver tomato was the first. People thought that in case of tomato, uh, shelf life is a big issue. So they developed flavor saver tomato, higher shelf life, but it was not, popular enough. But later, lots of, uh, uh, genetically modified crops being developed, and they are playing big role in global food security. Um, but in early stage, the biggest challenge of doing the genetic engineering was the uh, uh, unavailability of the defined gene. So genome sequences to decode the genome of the organism has been started in around 24 years ago. Arabidopsis, the first model plant, genome was sequenced, and the rice genome sequenced into 2002. And gradually, genome sequencing and molecular biology of different molecular biological work helped us to get the new gene responsible for or regulating the new trait so that we can design a new uh, breeding strategy for uh, developing the more climate smart or stress tolerant high yielding variety. Uh, in 2002, another breakthrough you can then gene finger nucleus for genome editing. It was the first, genome editing technology. It was a dream when James Watson and Francis Crick uh, proposed the double helical structure of DNA. At that time, people thought one day we can edit the genome and change the genetic trait and make the designer the organism. And it came true in 2002 when zinc finger nuclease for genome editing been discovered. And then another genome editing technology, 2011, transcription activator like effector nuclease. There are two genome editing technology we shall discuss later on. And the 2012, the are most revolutionary. That means people believe that in this century, clustered regularly interspace short palindromic repeats, uh, that is CRISPR-Cas system, is the greatest technology which won the Nobel Prize in chemistry, can help us to uh, design a new plant, new climate smart plant very easily. Other two technologies, zinc finger nuclease and transcription activator like effector nuclease, they are also genome editing technology, but they are cumbersome, difficult to do. But CRISPR-Cas system genome editing is really the sharpest technology to edit the DNA of an organism, and it is now uh, impacting to the agriculture, food security, as well as the human health and other purposes. And genome sequence of different organisms gradually being done. Now thousands of plants genome being sequenced, but genome sequencing cannot give you all the stories hidden in the secret black box of the DNA. So to decode the gene is really a challenging issue. Another challenging issue is that single gene uh, usually not regulate or 100% control a particular trait. So there are, gene when expressed, they produce protein. And in a cell, in a given time, lots of proteins are synthesized. They have the huge interaction. So it is not linearly regulated. This is why uh, editing or, modifying the genome is really challenging. We have to do lots of, work to figure out and get the desired plan. I mentioned before that the uh, breeding, now molecular breeding, we are at the age of genome editing. They are really a art, and you need, more insight about the functions of different genes, how they interact with each other, and without having uh, all the insights, you cannot make a big revolution. For example, this year Nobel Prize in physiology and medicine was given for the discovery of microRNA. MicroRNA, small RNA, which cannot, translate into a protein, but they can interfere the uh, 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 transcription and translation process of other genes, neighboring genes, or even other genes in the genome. So if you do not know how your gene expression, particular defined gene expression is regulating, you cannot make smart engineering. So I remember these two greatest uh, art. One Pablo Picasso, The Girl Before a Mirror, and another Leonardo da Vinci's Mona Lisa. And both I have seen, visited the museum, and they are, really, really a greatest art and give you lots of, insight, the power of art. Mona Lisa, from any angle, if you see, you will find that the lady is looking you. So, really amazing how it was made. And in science, complicacy may be similar to make something great and making the impact. Editing or genetic modification nowadays no problem, but to get the outcome uh, that means the improvement of the trait as well as climate smart variety development really still challenging. So breeding methods over the year, that method saves time, any method. So breeding methods should be faster. As regard his method, Aristotle is recognized as the inventor of scientific methods because of his refined analysis of logical implications contained in demonstrative discourse, which goes well beyond natural logic and does not owe anything to the ones who philosophized before him. Actually, Aristotle, uh, he is the father of politics, even father of biology, and they are, gave us lots of philosophy. In case of, life science, whenever you want to discover something new, you need to have some hypothesis, and then you can test it through rigorous methods. So, uh, foundation of science greatly, indebted to Aristotle and some great philosophers. Speed breeding, let me start this topic. Speed breeding, actually, this concept uh, officially, popularized by Dr. Lee Hickey, a young professor in Australia. But to speed up the breeding uh, was a target for centuries of the scientist. What he did, he stimulated the speed of plant growth by changing the light as well as other environment. He has been working in at University of Queensland. Very interesting guy. So he changed the environment and life cycle of, for example, wheat and other crops. He reduced significantly due to the changing the photo periods that are critical for the accelerating the flowering process. So if you, for example, in Bangladesh, we can grow wheat in winter. Now we are growing, but every year once only. But in case of rice, we can grow three crops. But in the control environment, if we can grow four wheat per year, then the breeding cycle you can accelerate fourfold. That is 4 years, the job you can do it now 1 year. So this is really great idea and a smart idea. What is speed breeding? Technology that saves breeding time and resources through very rapid generation advancement in a control environment. So that I mentioned. And results in 3 to 9 generations per year compared to 1 to 2 generations per year achieved with conventional selection approaches in the field. Here you can see the how changing in the different spectrum of light reduce the uh, lifespan uh, by accelerating the flowering, inducing the flowering, and reduce the breeding time. Uh, so in case of breeding, generation to generation, we have to observe it. So here variety release in the past it was uh, 10 years or even 15 years, but by using uh, this smart technology, where 3 to 9 generation per year we can reduce the variety release time significantly, even 3 years or like that. And this technology is really, an innovative thinking. And Rationally for speed breeding, already I discussed, but let me focus or reinforce it again. The world's population is expected to increase by over 20% over the next 30 years. That is, by 2050, it would be 9.7 or nearly 10 billion. By then, we will need to produce 974 more calories per person per day. That is, we need huge, food. Increased water use and elevated temperature caused by the climate change will negatively affect the food production because water scarcity, that is fresh water, really a big issue because for crop production or human and other animals, fresh water is a very big issue. So water supply is also a very big challenge. Speed breeding can help to increase food production by increasing genetic gain. Genetic gain means, different plant has different genetic traits. So the some plant may have insect resistant gene. So but if you can identify that gene, clone that gene, and picked up that gene and introduce into your commercial cultivar, then you can increase the yield, and we can call it genetic gain over time, and producing crops that are acclimated to new environmental condition. Climate smart, that is, we are now trying to develop uh, the new crop variety by using different genetic modification uh, so that they can adapt to the changing climate. Even flooding, plant can stay intact even 7 days or 21 days under the water or like that. So varieties are now scientists trying to develop. And there is another, I shall talk about it later on, double haploid production. Double haploid production. This is also can reduce the breeding time. So, and also gene editing is also included in the speed breeding process so that we can reduce the time for uh, producing the new variety. So, major challenges in Bangladesh food security already I discussed with you. We are adding in Bangladesh every year 2 million people are adding new mouths are adding to the our population, and the population increase by 2050 is expected to be 230 to 250 million. And we have a very small area, that is 8,000 that is 8.774 million hectares of cultivable land. Incredibly, small area. Our land area, total country area is, smaller than California, a single state of US or like that. Loss of 1% of agricultural land annually. I mentioned this point earlier. So additional challenges are biotic and abiotic stresses, salinity intrusion in coastal belts, and they are coming toward the north, chronic and seasonal drought, limited organic matter. Organic matter is called the, life of the soil. Organic matter is decreasing and it should be more than 2% for agricultural soil. But in Bangladesh, it is in most cases less than 1%. And post harvest food loss is another big issue. So Bangladesh must develop customized, improved, and elite crop varieties that can address these challenges in a short period for smallholder farmers in 5 to 6 years instead of 10 to 12 years through the traditional breeding. This is why these are all rationally of the speed breeding. In case of conventional breeding, you can see crossing and inbreeding 3 to 7 years. Testing in the field takes 4 to 5 years, and then seed increase and release to the farmers. That means it takes 10 years or more to get the variety in case of wheat or rice even. But if you go more detail about the conventional cycle for breeding, here you can see how we do it. For example, parent 1 and parent 2, if parent 2 carries some important trait, we cross and then we get the F1 then segregate and then gradually selfing and back crossing, we can get a trait settled in the targeted crop, and then we go for the field testing and varietal development and release the variety. So this is the conventional cycle time for breeding and the pathway and illustration how we do it. So it takes 10 to 15 years depending on the crop. And breeding time, as mentioned, uh, some of the new breeding technologies being developed to shorten, for example, shuttle breeding. He is the, Norman Borlaug, I mentioned before, the Nobel Prize winner. He visited Bangladesh. So shuttle breeding sometimes can reduce a little bit, but even though take a long time. But if you go for the speed breeding, you can save the cost, save the time, and accelerate the genetic gain to solve the problem. In speed breeding, not only changing the light and other environment, but also inclusion of double haploid technology. Double haploid technology I shall discuss later on. How can we get the pure line very shortly? Because if uh, homozygosity is not achieved, then uh, the stability of the developed variety would not be achieved. So it helps us and save time, as well as computational modeling we can use and predict if we deploy this gene how it would impact by using bioinformatic analysis. And CRISPR-Cas genome editing is saving huge time because it is very precise precision breeding. So uh, combining all the smart tools, the breeders are now making new strategy to get the variety with uh, new trait within the as much as possible of the shortest period of time. So I would like to take a little break and allow you to ask few questions. So it is your time. Please ask few questions. Ask me a question, whatever a question in your mind. Hello, I am audible?
Yes, sir, you are.
Excellent. So your name is Rian?
I'm sorry, Ryan.
Ryan Mehjabin. Okay. Uh, please ask a few questions so that I can sense that you are getting the lecture rightly.
Sir, no question so far because trying to understand what you have said. This
Hmm.
is completely new thing for me.
Hmm. Obviously, because agriculture, is not was not measured in your undergrad. This
Yes,
is
sir. Yes, sir.
why lots of things you will, find new. But obviously, as the topic is directly related to your food that you are getting on your dining, or uh, every day you are getting the food. So uh, I think you can sense it. And Bangladesh is basically uh, uh, an agrarian country. So some all of we have the root in the village. The growers are not alien for us. They are our own people. And the breeders, and other scientists, they are relentlessly working. And over the years, after the independence, Bangladesh, uh, you know, uh, made significant success in increasing the food production. We are almost free from famine and hunger, but still big challenge of getting food in reasonable price for all the people all the time. That means food security, high quality food, and the availability and affordability, as well as you know the safety, very important. Do anyone have any question? Please ask a few questions.
Sir, can you explain the shuttle breeding again?
Shuttle breeding, yeah. Shuttle, uh, shuttle bus, or shuttle in our transport system. So once, uh, the breeding actually, what we do classically, we cross to dissimilar parent, as you have seen here, to get trait from one parent to another. Artificially, we do crossing very precisely so that no interference are taken place. And then the F1 segregate into different populations. So the seed from F1 will give different, phenotypes. So you picked up your, desired phenotype, and then do selfing again, and gradually you move on, and it needs, at least 6, 7 generation to get nearly pure homozygous or isogenic you line with the trait. So lots of undesirable one you will discard. But in case of shuttle breeding, uh, the breeding process I mentioned, it is linear. But in case of shuttle breeding, you can grow 2 generations per year. It was, thought that if we can grow 2 generations per year, then we can reduce the breeding time half or like that. But it depends on which crop you are handling. So uh, in case of rice, no problem. In case of some other crops, short duration crop, no problem. But if you go for the sugarcane or some other cotton, not so easy. But shuttle breeding parallelly you do the two generation, that is two generation per year. How can you do it? There were some smart technique, to deprive the plant for nutrition. So, less nutrition can give the flowering earlier. Your intention is to make a shorter lifespan for getting seed to seed, and then uh, even a year you can get two generations. So you can shorten the uh, how to say the total breeding time. But that were different other tricks are also done. But nowadays, no need shuttle breeding. We have now more smarter, that is speed breeding, as well as combined with the double haploid. Double haploid means you culture the pollen grain on the plate, tissue culture, and get the haploid plant because it is not fertilized. By using colchicine, you can make it double haploid. And double haploid can give you the flower and other things. So these are the double haploid technology I shall tell you later on. But a shuttle breeding in brief is that uh, by using different environment and get the uh, two generation per year uh, for us, speed up the breeding technology. Uh, do anyone have any more question? If not, then I shall move a little bit more. So we already understood, I hope, the speed breeding technologies. The objective is to shorten the breeding cycle. And speed breeding technology, here you can see how we can develop the uh, rapid, uh, how rapidly we can uh, stack the gene by crossing, and even different donor plant we can use to uh, uh, you know uh, incorporate a gene and get the variety. You know that in case of genetic engineering, agrobacterium mediated transformation, back crossing, back crossing means when we do, these are donors contain the different, traits. Here you can see different gene, this gene, this gene, this gene, this gene. They are different. Some are disease resistant genes, some are, drought tolerant or some other ill related. So your elite variety, it has not such special trait. So what you need to do, the cross, and then do the back cross. So back crossing means the F1 you cross with the F2. You cross with the parent elite variety continuously, and finally remove the all undesirable traits and stack several genes together. For example, here 4 genes you are intended to introduce into a variety. So back crossing 6 times or like that, you will get the homozygous plant and a new variety. But in case of speed breeding, a classical process, it takes a longer time, you see 8 years. But in case of speed breeding, 3 years can be possible because you can speed up the process. Here, double
Sir,
head.
I
Yes,
have a question
yes.
in this slide. স্যার F1 এ এটা আমি প্রথম মিড টার্মের ক্ষেত্রে একটু কনফিউশন ছিল। F1 এ 50%, এটা কি স্যার 50% হচ্ছে এলিট ভ্যারাইটি আসছে, মানে আমি এটা 75% কি বোঝাচ্ছে, এটা একটু যদি ক্লিয়ার করে দেন।
হ্যাঁ, আমি এটা বলছি, যখন আমরা F1 দুটো ভ্যারাইটি, এলিট ভ্যারাইটির সাথে ডোনার প্লান্ট, যেমন তুমি একটা জিন ইন্টিগ্রেট করেছিলা জেনেটিক ইঞ্জিনিয়ারিং এর মাধ্যমে, করার পরে যে প্রথম প্লান্টটা হয়েছে তার মধ্যে তোমার যে নতুন জিন প্রবেশ করেছে, এই জন্য এটার পরের জেনারেশনে মেন্ডেলিয়ান ল ফলো করে সেগ্রিগেশন হবে। এটা সবগুলো তার প্রজেনির মধ্যে এই জিনটা স্টেবল থাকবে না, কোনগুলোতে স্টেবল থাকবে আর অনেকগুলোতে বিভিন্ন রকমের কম্বিনেশন এবং ইন্টারাকশন হবে। তো তুমি এখানে নিয়ারলি 50% প্ল্যান্ট পাওয়া যাবে যেটাতে ওই জিনটাও আছে, আবার এলিট ভ্যারাইটির ট্রেইটও আছে, যেটা তুমি এক্সপেক্ট করছো। তো এই জন্য এটাকে যদি আমরা ব্যাক ক্রসিং করি, যে এই F1 এর যে প্রজেনি হয়েছে প্যারেন্টের সাথে, এবং এইভাবে যদি করতে থাকি, তাহলে একটা পর্যায়ে গিয়ে তুমি এলিট ভ্যারাইটির ফুল ট্রেড থাকবে, প্লাস হচ্ছে নিউটা পেয়ে যাওয়া, এবং এটা স্টেবল। এরপরে এই সিড যদি তুমি ফার্মারকে দাও, কমার্শিয়ালি প্রডিউস করো, এটা আর চেঞ্জ হবে না, ফিল্ডে গিয়ে সেগ্রিগেশন হবে না। আমি তোমাকে বলেছিলাম যে যখন কোনো প্রোটিন তৈরি হয়, ওই প্রোটিন অ্যালোন লিনিয়ারলি এক্সপ্রেস করে না, এখানে একটা স্ট্যাবিলিটি তৈরি হয়। এই স্ট্যাবিলিটিটা, এই ইন্টারেকশন গুলো 100% কার সাথে কে ইন্টারেকশন হবে কম্পিটিশনাল বায়োলজির মাধ্যমে আমরা কিছুটা জানি, কিন্তু একটা প্ল্যান্টের মধ্যে 30,000 40,000 জিন আছে। কাজেই এটা রিয়েল ফিল্ডে এই একটা জিনিস জানো, শুধু জিন থাকলেই হয় না, এনভায়রনমেন্ট একটা বিরাট ফ্যাক্টর, যে এনভায়রনমেন্টে এটা চাষ করছে। তো ওখানে গিয়ে একটা স্ট্যাবিলিটি যখন আসে, তখন আমরা এটাকে ওই জিন স্ট্যাক অথবা জিন ইন্টিগ্রেসড অথবা জেনেটিক্যালি মডিফাইড এই ভ্যারাইটিটাকে আমরা রিলিজ করতে পারি সিড আকারে। তখন এরপরে যখন এটা চাষ করবে, এটা স্টেবল হয়ে যাবে, এবং এটা তোমার ইউনিফর্মলি সব প্লান্ট যখন 96.87 অথবা 99% তোমার হোমোজাইগোসিটি চলে আসবে, অর্থাৎ এই জিনের মধ্যে যে রিঅ্যারেঞ্জমেন্ট হয়েছিল এটা সেট হয়ে যাবে। আমরা ইনফ্যাক্ট এক্স্যাক্টলি কোন লোকেশনটাতে জিনটা অ্যাড হবে, এগ্রোব্যাকটেরিয়াম বলো আর ক্রসিং বলো, অনেক ক্রোমোজোম থাকে, এটা কিন্তু প্রিসাইজলি আমরা একেবারে সুনির্দিষ্ট করা খুব কঠিন, ইভেন এডিটিং এর মাধ্যমে। ফলে এটা এই বড় রকমের এই নতুন গ্যাস্ট জিন যে গিয়েছে, তার সাথে নেটিভ জিনের অনেক ইন্টারেকশন হয়, জিন প্রোডাক্টের ইন্টারেকশন হয়। তো এই স্টেবিলিটিটা যখন আসে, তখন আমরা এটাকে ভ্যারাইটি হিসাবে রিলিজ করি, এবং এইটা তখন ইউনিফর্মলি ফিল্ডের মধ্যে গ্রো হবে, গ্যারান্টি দিয়ে তুমি ইল্ড, অর্থাৎ যে ট্রেডটা দিয়েছো ট্রেডের বেনিফিট পাওয়া। এটা কি ক্লিয়ার হয়েছে?
জি স্যার, থ্যাংক ইউ, আমি বুঝতে পেরেছি।
Now I would like to talk a little bit about double haploid. Double haploid, here you can see the process. Uh, Double haploid is a technology to get the homozygous plant. How can we do it? Here you can see we start with the pollen. Pollen grain. So you can get the anther. Here you can see isolated anther. When a flower is blossom, you can find in the stamen lots of haploid anther, and they are basically through the wind or insect transported to the stigma, the female sex organ of the plant. So anther, if you culture in the medium, like tissue culture. Anther culture is one kind of tissue culture. So you can find these scalars proliferated on the medium. So anther cell profusely grow and make multiply. And then by changing different hormone, you can generate the, haploid callus. That is, you can see here the greenish. And then you can generate the shoot. You see, shoot differentiation, haploid plant, like tissue culture we do. And then what you do, by using colchicine treatment, you can make them double haploid. Mane chromosome number doubled. And then you can get the double haploid plant. As there is no mixture of any genetic, material through the crossing and others, they are pure homozygous plant. And uh, to get the pure homozygous plant, as I mentioned in back crossing, it takes time. But here, very quickly, you can get the pure homozygous plant. And in the breeding technology, people are now using this technology. After the editing, if you get this type of job, then you can get easily pure double haploid plant, homozygous plant, and that save the breeding cycle. And here you can see how pollen is collected and cultured. That is pollen you can culture in the pollen you can anther, and pollen you can culture in the tissue culture medium, and then you can get the haploid plant. And pollen are free from any contamination. So this is really interesting method, and it is called male germline. That is embryogenesis. All are male germline. From the male germline, you can, if you get the haploid plant, it would not flower. But if you make them double haploid, then it would be homozygous. And it is widely employed in many row crops. Strong plant to plant variation requires optimization of culturing condition. Perfectly pure, fertile, diploid plant can be generated in a single generation. Here, a haploid plant is infertile and useless. But here, a double haploid, fertile, and you can do any other job, even gene editing and modification, again, because they are stable, double haploid, pure line, and good for breeding. And if there is in the genome, if there is heterozygosity, then to get a pure breeding line to release the variety is really difficult. So in case of breeding, for example, genomic selection optimizes the breeding values based on the incremental value of each genomic segment. When you do any breeding, every plant we care, because which plant is giving you the desired trait. Although theoretically, when you introduce a gene, all plants should behave exactly the same. But I mentioned exactly not, and gradually they segregate, and finally we find the best plant. So best plant we can select, and then by visual observation, as well as we can sequence the DNA and see whether when you add a gene, sometimes it can affect the expression of other gene and can cause the pleiotropic effect, depending on where gene is added in the genome. So this is why you need to do some, selfing as well as back crossing to get the stabilize your trait with your desired character, and visually phenotyping as well as genotyping, DNA sequencing also sometimes we do, and to see the where it is incorporated. So optimizing breeding pipelines, lots of factors are involved. For example, rapid breeding cycle, high selection accuracy, selection of parent with high breeding lines. Nowadays, imaging technology, drone and other technology are being used to get the phenotypic data, not one by one you need to take. And there are software you can pick up the plant, not manually, using the software, using the unmanned vehicle like drone. And breeding cycle we can change by using different physico-chemical condition, lighting and other things, and reduce the, even adding the double haploid technology. Even in the breeding we can add the CRISPR technology for reducing the time. So supporting speed breeding, uh, uh, I visited, the Global Institute for Food Security. They are using omics and precision agriculture, data management, analysis, cell biology, plant growth, and all the things, genotyping, phenotyping, speed breeding, and genomic selection. All they are incorporated to make the uh, crop improvement more precise and get the desired results very quickly. If I ask you what are the major advantages of the speed breeding, the most important advantage, number one, is shortened breeding cycle time. Number two is genomic selection considers performance of lines across multiple environments. That is, breeding values incorporate performance of crops in current environment, the environment we are using, because environment are critical factor for regulating the expression of the gene. Third, new parental combinations based on complementary breeding values. Third, visual characterization. Time is reduced by discarding lines with low breeding values because people can use unmanned vehicles or like this. And breeding pipeline is modeled to identify most efficient practices. So several advantages are there. Here you can see speed breeding and glass house control. So here uh, you can see how speed up can be done. Wheat, barley, chickpea, canola, people used in the controlled system, and they reduce the time for uh, significantly reduce the time for breeding, and lots of varieties have been developed by using this speed breeding technology. And another interesting point is that harvesting immature spike. That means your target is how shorten uh, you can make the breeding cycle. Uh, you know, 15 days, for example, after the anthesis, that is when seed is formed, but seed is not ripened yet, you can harvest. And seed maybe shrink. Then you use the 35 degrees Celsius to dry the seed. Because you can even save 1 week for the breeding cycle. Here, seed is shrink, but does not matter. If it germinates, then it is enough to get the new generation, because genetic makeup is not changed if it is, shrink seed not fully ripened. So lots of smart approaches people are using to get the speed up the breeding cycle. Here it is called speed breeding to generation. That means reducing operating cost. They are using LED light. So LED light is less cost effective. John Innes Center, I visited that one in UK. They have these speed breeding facilities. So boosting transgenic and CRISPR pipelines. Also, we can do the speed breeding, because when you do the transformation, agrobacterium mediated for editing or others, then you can go for the speed breeding for stabilizing, as well as you can go for the double haploid for getting the homozygous line very quickly. So opportunities, commercial breeding companies using speed breeding need to invest in infrastructure and outsource service, because you need to have such a sophisticated, controlled environment, no electricity failure or no interruption. So initial cost here involves, as well as the other infrastructure. Can we take speed breeding off the grid? No, it is possible, but you need other generator. Winter wheat still requires vernalization. Yes, some of the winter wheat, possibly you know that in case of temperate country, they sow the seed of wheat before the winter, and then the seedling a little grow and snow covers them for several months, and it is treated by the cold treatment called vernalization. Vernalization means cold treatment to allow the plants, wake up the plants for flowering. So winter wheat, without vernalization, if you grow in Bangladesh, you cannot get the flower because naturally they have the system. So what we need to do, the seedling, after germination, you need to put your refrigerator for 35 days or like that. Then if you grow, you will get the flower or like that. So vernalization in case of winter wheat is required. You cannot shorten it. Speed breeding, bigger gains likely possible through program redesign, opportunity to combine speed breeding as well as CRISPR to get the real speedy outcome. And the CRISPR technology and genome editing, I shall discuss tomorrow. Now, it is time for you to ask some question. In case of CRISPR, these two lady, Emmanuel Charpentier and Jennifer Doudna, they got the Nobel Prize in Chemistry in 2020, and they developed this technology that is to cut the double-strand DNA using the synthetic guide RNA with the CRISPR construct, and CRISPR contract is molecular scissor and cut the DNA in the desired location as you synthesize the guide RNA, complementary sequence of your target gene. And if it cuts the DNA, then DNA has inherent properties to, repair. And during repairing, some gene may be, one or two nucleotide and few nucleotides may be missing. And if it is a bad gene, it can be inactivated, and this deletion disrupt the expression of the gene. And if it is your desired, target, then it will work without adding anything. Or you can put if there is a wrong sequence, you can put a template, and in the cut point, it would be, incorporated, and you can make a correction if there is any wrong sequence due to mutation. So gene has a new sequence, or even new gene can be incorporated. So really, really smart. Not only this, at nucleotide level, gene editing using CRISPR or prime editing or base editing, they are becoming and evolving very fast at the specific epigenetic level, or a single base, you can edit and make the change. So I shall discuss tomorrow detail about the genome editing, how it is becoming a robust technology for rewriting the biology, and especially in genetic modification or manipulation of the plant for ensuring the food security is advancing. One important point is that although CRISPR as well as genome editing, both scientists, they are not agriculturists, plant scientists. Emmanuel is a microbiologist and Jennifer is a biochemist. Their ultimate target is to apply in human. That means editing the human genome for curing the cancer and other, genetic diseases. Their dream is becoming true because sickle cell disease, due to a single mutation, wrong mutation, it is a big sickle cell anemia is big problem. By using CRISPR-Cas genome editing, it has been now changing the life of many people in US, and it received the permission for real life use. So I shall discuss in detail how CRISPR is making a new revolution in the life science, and I shall focus its application in the plant and agriculture, because application of any such technology in plants are easier for many reasons I discussed before. Doing research with a plant you have less ethical obligation, and can life cycle is short, plant is pluripotent, transformation is easy. So the impact of genome editing uh, is making bigger impact in agriculture and food security. Thank you from my side, and you can ask now some question. It is your turn to ask some question. So 25 students, I can see 24 students are now attending. If you have listened correctly and without any interruption, I expect you will ask few questions.
স্যার আসসালামু আলাইকুম স্যার।
Yes, Abrar.
স্যার একটা কোশ্চেন ছিল স্যার, এই যে ডাবল হ্যাপ্লয়েডস প্রোডাকশনে স্যার আপনি মেনশন করছিলেন অ্যান্ড্রোজেনেসিস মেল জার্মলাইন। সো মেল জার্মলাইন স্যার এখানে কেন ইউজ করা হয়?
এটা খুব ভালো প্রশ্ন। মেল জার্ম লাইমস অর্থাৎ পোলেন, পোলেন গ্রেইন অথবা তুমি অ্যান্থারকে কালচার করে আমরা এই টিস্যু কালচারের মাধ্যমে অ্যান্থারকে কালচার করে আমরা হ্যাপ্লয়েড প্লান্ট পেতে পারি। এখন এটা কেন করা হয়? ফিমেল যে অর্গান এই মেইলের মতো এইরকম ইউনিক না। মেইল পোলেনটা তুমি যদি দেখো বা এন্থারটা, এটা একেবারেই তোমার অনেকটা স্পার্ম সেলের মতো হিউজ অ্যাবান্ডেন্ট, তুমি এখান থেকে নিতে পারো। কিন্তু ফিমেল যে ফ্লাওয়ারে যে সেক্স ওরান্ট একটা স্টিগমা, এটার ভিতর থেকে অভিউল নিয়ে এটা ভেরি কমপ্লিকেটেড। আই এম নট এক্স্যাক্টলি শিওর, তবে এইটা খুব ইজিলি বোঝা যাচ্ছে। পোলেন গ্রেইন তুমি হাত দিয়ে টাচ করলে একটা ফ্লাওয়ারে দেখবা থাউজেন্ডস ইভেন মিলিয়নস অফ গ্রেইন ইন্ডিভিজুয়াল, অ্যান্ড দে আর পিওর সেল অ্যান্ড ইজি টু কালচার। এটা অনেকটা মেরিস্টিমেটিক টিস্যুর মতো, যেমন এটাকে কালচার করে তাদের মধ্যে ওই যে টটিপটেন্সি এটাতে থাকে, এটাতে কোনো প্যাথোজেন থাকে না, প্যাথোজেন ফ্রি, ভাইরাস ফ্রি, ফলে এটাকে কালচার করে নিউ প্লান্ট পাওয়া অনেক ইজি। পোলেন কালচারটা অনেকটা সিডের মতো এটা করে, কিন্তু যখন এটা তোমার হ্যাপ্লয়েড হয়, এটা কিন্তু স্টেরাইল, একা শুধু পোলেন, এটা তুমি সিড নতুন তৈরি করতে পারবে না ফ্লাওয়ারিং দিয়ে। সেই জন্য কলসিসিন দিয়ে যখন ডাবল হ্যাপ্লয়েড করা হয়, তখন এইটা তোমার ফার্টাইল হয়ে যায়। কিন্তু হোমোজাইগাস, কোন রকমের এখানে ভেজাল নেই, যেহেতু সিঙ্গেল সেল থেকে এটা করা হয়েছে। ক্লিয়ার আবরার? থ্যাংক ইউ। আর কারো প্রশ্ন? খুব ভালো প্রশ্ন করেছ। আর আমি ডাবল হ্যাপ্লয়েড নিয়ে আরেকটা স্পেশাল লেকচার দিব, যাতে আরো একেবারে ক্রিস্টাল ক্লিয়ার হয়। আর কারো প্রশ্ন? দু একটা প্রশ্ন করো আরো। আরো 5-6 মিনিট আমি চাই যে একটু ডিসকাশনে আসো। ডিসকাশনে আসলে এটা ক্লিয়ার হবে, কারণ আমার জব তো মনে করো যে লেকচার দেয়া, কিন্তু তোমার জব হচ্ছে এখানে যত বেশি ইন্টারাকশন করবা, সলিড আন্ডারস্ট্যান্ডিং হবে। সবাই কি শোনা যাচ্ছিল, বা কেউ কি কোনো ইন্টারাপ্টেড হয়েছিল যে ইলেকট্রিসিটি বা অন্য কোনো কারণে? না।
জি স্যার।
কি সঞ্জিদা?
মাঝখানে নেট ছিল না স্যার, পরে আবার জয়েন করেছি।
আচ্ছা, প্রশ্ন আছে কোনো?
জি না
আমি
স্যার।
আমি ক্যাম্পাসে এই উইকডেতে না থাকার কারণে তোমাদের হয়তো অনেকে মিস করো ইন্টারাকশন করতে। ইন্ডিভিজুয়াল ইন্টারাকশনের জন্য যদি ইমার্জেন্সি কোনো ইস্যু হয় তখন হোয়াটসঅ্যাপে কল দিতে পারো। আরেকটা হচ্ছে আমি কালকে একটু আর্লি যাব, যাতে আহ, এই যারা কোনো টপিক ভালোভাবে বুঝতে পারো না, ডিসকাশন করতে চাও, ঘন্টাখানেক আগে আমি যেতে পারি, তখন ওখানে এসে ডিসকাস করতে পারবো। ওভারঅল অন্য সাবজেক্টের তুলনায় এটা কিরকম ফলো করা যাচ্ছে, নাকি খুব কমপ্লিকেটেড মনে হচ্ছে? ইমপ্রেশন কেমন তোমাদের? এইটা তো প্রশ্ন না যে একবারে তোমার সাবজেক্টের, তুমি এটার তো কোনো না কোনো আনসার আছে তোমাদের কাছে। হুম।
No, sir, so far so good.
So far so good.
But it should be more edible, sir. We should say.
Great.
Because some terms are not related with our past educational background. Sir, if you take it in your concern, that would be very helpful for us. Thank you.
Obviously, in the first class I asked uh, you to introduce yourself, and I tried to get your background. This is why uh, I am moving very slowly and trying to give you enough time for interaction, because here the whole group is highly heterogeneous. Many of the keywords or terms are new. Thank you so much. You really mentioned important point. Yes, I am always caring of it, but as I have been uh, trained not only in plant science, I received training in microbiology, chemistry, plant biology, genetic engineering, lots of things, because over last 30 years, uh, 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 my background is quite versatile. And if you search my website, you can find publication in coronavirus, COVID-19. You can find publication in fisheries. You can find publication in microbiology, obviously plant biotechnology, genetic engineering, chemistry. So for me, if you interact well, then for me it would be uh, uh, delightful for me to uh, share and analyze the topic uh, as palatable to you. Do anyone have other question? Okay, then thank you so much, and I shall try to save the recording. If it works, then I shall share it through the YouTube, and you can visit it again. Thank you so much. Have a good day.